Shall we start now, sir? So very good afternoon. I hope you, let's welcome to the, we welcome you to the 6th technical session of the 3-day international conference on biotechnological applications in Ayurveda and biomedical sciences and the annual meet of the Society for Biotechnologists, SBTI India, organized by the Faculty of Allied Health Sciences and Society for Biotechnologists, India. This is an interesting session where we'll be delving into topics intersecting biotechnology, microbiology, food security, and nutrition. The session promises to bring forth exciting new perspectives that are shaping the future of scientific research. To moderate the session, we are privileged to have with us Professor Dr. Rajeshri Gaur sir as the chair and Dr. Rajkumar sir as the co-chair for the session. Professor Dr. May I request sir to kindly, we welcome you sir. Professor Dr. Rajeshri Gaur sir is a leading expert in plant virology and biotechnology. As the head of the Department of Biotechnology at Deen Dayal Upadhyay Gorakhpur University, sir is also the Coordinator Research and Development Cell, DDU Gorakhpur University. Sir's groundbreaking contribution to the field includes 28 reporting of the plant viruses for the first time in India and pioneering RNAi methodology for resistance against virus. Professor's expertise is widely recognized through his editorial roles in several reputed journals such as Indian Phytopathology Springer, Frontiers in Microbiology, and BMC Plant Biology. Sir's dedication to education and leadership has been recognized with several prestigious awards, including Member of National Academy of Sciences, India, Best Professor Pearl Foundation, Professor B.M. Johari Memorial Award, Society of Plant Research, Excellent Teacher Award by Asta Foundation, INSA Summer Research Fellowship, UGC Research Teacher Award, and many more. With his remarkable contribution to science and research, we are privileged to have Sir leading today's session. Please join me in welcoming our chairperson for the session, Professor Dr. Rajeshri Kumar Gaur sir, with a big round of applause. It is an absolute honor to have with us Professor Dr. Rajkumar sir as our co-chairperson. Professor Dr. Rajkumar is the professor in the Department of Biochemistry, BRD Medical College, Gorakhpur. Sir has more than 20 years of teaching experience and several research papers and books to his credit. We welcome you, sir. Kindly welcome, sir, with a big round of applause. We have Dr. Prerna Aditi, faculty, as the rapporteur for the session. This session promises to be an engaging one with eminent speakers being lined up. So we have our first presenter today, Professor Dr. Sarita G. Bhatt, Madam. Ma'am serves as a professor at the Department of Biotechnology, Cochin University of Science and Technology, with more than 25 years of teaching and research experience in biotechnology and microbiology. Ma'am's areas of expertise lies in bacteriophage therapy, gut dysbiosis and health, gut microbiome, probiotics, and its application. Ma'am is before us to present a lecture titled Gut Microbiome and Gut Health, Bacillus amyloliquefacins as the new probiotic on the blog. Please join me in welcoming Professor Dr. Sarita G. Bhatt, Madam, with a round of applause.
Thank you, sir. A very good afternoon to one and all. Thank you so much for having me here today. It's a great honor to be invited to talk at this international conference, which combines two different, medicinal approaches, the Ayurvedic and the allopathic medical practices. And we are also looking at medical or biotechnological interventions. I'm greatly honored to be here, and I'm going to talk about what we have been doing in our lab for the last 10 years or so. We have been working on the gut microbiome, and the topic today is gut microbiome and gut health, where we are going to take a look at Bacillus myeloliquefaciens as a new probiotic bacteria. So what we have seen is something that started with the Human Genome Project, or a little earlier than that, when Sanger sequencing was put forth. So there was a great interest in understanding the genetic makeup of both pathogenic and useful bacteria. We also understand during that process that there were projects like the Gut Microbiome Project or the Human Microbiome Projects, which helped us to understand about the microbiome in which we exist. We understood from this particular project and the associated or allied projects that humans have more bacteria on them and in them than there are human cells. We understand today that we have more than 10 raised to 4 microbial organisms, which includes both viruses, bacteria, as well as other microscopic organisms. We are more microbiota than human cell, because 1 in every 10 cell is only a human cell. So that is how interesting we should be about what is happening with us. So we'll just take a look at some terminology. The microbiota indicates the collective microbes that are present in and on the human. The microbiome is the genetic makeup of this entire human microbiota. Dysbiosis is where there is a change or a shift from the normal microbiota that is present. And we are going to take a look specifically at the gut. It's very easy for me today because the speakers this morning who spoke about Ayurveda, Dr. Tripathi also spoke about Ayurveda and how it impacts us. What we understand is everything that happens in the human being finds a way or a route through the gut. We are what we eat, and there are also sayings that have been ascribed to say that let thy food be your medicine and not thy medicine thy food. So we have so many things like that. वैसे भी बोलते हैं ना, जैसा अन्न वैसा मन. So all of these are related, and there are scientific proofs to prove that what you eat can affect not just your general health but also your mental health. So what is the gut microbiome? What is the human microbiome? What is the gut microbiome? What role does it play in health and disease? And is a modification of a human microbiome possible? Can we do this? Is it possible to treat, to maintain it and treat disease? So let's take a look at what this is. So we say that it is a very complex composition of bacteria, viruses. So actually that advertisement that comes in the TV very often, if you recollect, dhote jao, this is for all the kids who are sitting there. You have been told that you have to wash off all the microbes on you. Never happens. And if it does happen, you will be in great trouble, because there are these guys who are waiting in the wings to take over. Okay, but what we must understand here is that more than 99.99% of the bacteria that inhabit us are friendly bacteria. And it is only when there is a dysbiosis, due to various reasons, we'll take a look at that, is when this one among them becomes an opportunist, and that is where you have the onset of diseases. So what are the factors that affect the gut microbiome? All of us must first understand that we get our first microbiome. We are seeded with this microbiome when we are born. We get it from our mother when we pass through the vaginal passage during birth. Anybody who has a C-section would find that they do not get their parental microbiome, and therefore for their microbiome to stabilize, which even when you have a normal birth takes around 3 to 4 years, takes considerably longer time to do so. And that is when these kids would have large number of issues dealing with infections. And you can see that there are various things that can affect our microbiome: stress, the food we eat, the medicines we eat, the various aspects. So you can see that what we put inside of us affects us, what is present outside of us also affects us, because all of these can stress us out. So why is this microbiome such an important factor? Why do we say that it is so important to maintain this? This is because they do a large number of things for us. For example, they produce the small chain or short chain fatty acids. Anti-inflammatory production, they increase. They reduce the inflammatory mediators. They facilitate digestion. They produce vitamins. They ensure that there is a healthy gut epithelium. Mucosal immunity is enhanced. Body fat deposition is reduced, and so on. So there is a large number of advantages for us to maintain this healthy microbiome on us. So what are the disadvantages? We must understand that our gut, this thing that we have from our mouth to the anus, is connected to every single organ in our body through what is called as the gut-brain axis, gut-liver axis, gut-skin axis, and any other organ axis that you want. And all of this can impact. We have seen that there are a large number of issues that were spoken earlier in the morning about aging. One of these aspects is also aging, disease and aging. Okay? So you can see that any dysbiosis in the gut would cause considerably different types of disorders to come up, because these microorganisms play a very important role. And on this other side, we have listed a large number of gut microbiota strains. These are the strains that we look on as friendly bacteria. You may label them as probiotic bacteria, but doesn't mean that they are the only ones. We must understand that there are as many unculturable bacteria that we have on and in us as there are. There are more of unculturable bacteria than there are culturable bacteria. And these, because you can see their names here, you must understand that they are all being cultured and identified. So what happens to the gut microbiota? Let's take a look at inflammatory bowel disease and inflammatory bowel syndrome. Okay, so pathogenesis is linked to the inappropriate inactivation of this gut GI immune system. There is an enhancement of the immune system, and it happens more so in individuals who are susceptible, okay, and who have other factors also coming into the influence. So does the intestinal gut microbiota differ in patients who have specific disorders? And from the healthy individuals, we should say yes, they do, with considerable variation in the gut. So if the gut microbiota varies, what is it that is happening here? So what we must understand here is this IBD or IBS is a shift. It is just a shift. Your stress can cause a shift. The food that you eat can cause a shift. The medicine that you eat, like an antibiotic, can cause a shift, because all of them are influencing the way your bacteria can grow. So any shift from from all these factors will influence both your pro-inflammatory bacteria and your anti-inflammatory bacteria, which is balanced in a healthy individual. But what I must say here is that more than 10 to 15% of individuals world over have IBD or IBS. Okay, so this is something that we are battling with every day. So what happens? Why do we say that anything in the gut will affect every other tissue or organ? In our gut, we have what is called as these epithelial cells. Okay, so if we have what is called as leaky gut, everybody would have heard about leaky gut. Okay, this is when your gut epithelial cells, when these junctions, side junctions, they are disrupted and they open up. Okay, all the gut contents would leak out and they would go wherever it can possibly go and cause a problem. So a leaky gut will be damaged, will be lead to loss of membrane integrity. All your cellular contents that are present in the lumen of your gut, everything from broken down cells to other metabolites would all leak out. Okay, and it can go and cause an infection or a disease or a syndrome anywhere else. Healthy gut ensures that your microbial diversity is maintained. It ensures that and improves your intestinal barrier, ensures that there is reduced infection by the various pathogens that are present, and increases the. So what are the diseases that are associated with the gut? Inflammatory bowel disease. There are other metabolic disorders that we talk about. Infections and antibiotic use. Antibiotic use is often associated with dysbiosis, and it takes a considerably longer period of time for this microbiome to normalize. That is also therefore important for us to ensure that there is no on off the counter antibiotics that are taken by individuals on their own. Okay, then we have neurological and mental health disorders. We know today that anything that affects our gut also affects our mental health. This feeling of well-being that we have, okay, because there are a large number of hormones and neurotransmitters that are secreted in the gut. It's a considerably long organ. So there are a large number of them produced, and therefore they would certainly affect how you feel. So you have changes that are linked, changes in the gut microbiome, which is linked to the brain via the gut-brain axis. We have seen this, we'll just move along. So inflammatory bowel disease is because of leaky gut. You have a probiotic loss, you have microbial dysbiosis, but application of a probiotic can help you to give a large number of benefits, because your probiotic will help to strengthen the gut barrier, reduce immune reaction, and then reduce the leaky gut, improves the repairs, same thing all over again. Okay, I tried to use AI and this is what it gave. Okay, not very reliable. So what are the different types of treatments that are available for IBD or IBS? Okay, you have large number of treatment possibilities: salicylates, cortisols, immunomodulators, biologics, and you have antibiotics. Nothing novel about the fact that there is an antibiotic. Okay, it just went off. Okay, but we have prebiotics, probiotics, postbiotics, symbiotics. There's also what is called as fecal matter transplant, or FMT as it's called. We will not take a look at that. But all of these started with this guy way back in 1907, when he looked at bacteria and said that they were described as probiotic. Now we must understand that this probiotic has a large as a big market value, okay, to $21.6 billion global in 2010. It is $31.1 billion in 2015, and it is expected to go beyond 40 billion in another 5 years. So a lot of possibilities. Why? Because we are always living constantly in a state of stress. So these probiotics are live bacteria which are beneficial. In what way are they beneficial and where are they used? They can be used for all kinds of gut-related disorders, H. pylori eradication, when there are acute diseases that are associated specifically with infections like Clostridium difficile. Okay? Then lactose intolerance, and of course functional bowel disorders. So what are these probiotics? They are nothing but gut helpers, and because they have a large number of functions that are associated with them. Different groups have different types of functions. We have lactic acid bacteria, we have bifidobacterium, we have bacillus, which is a spore forming bacteria. We have the streptococcus, which are found in the yogurt and fermented foods. Now, what is the mechanism that is proposed, or what is the mechanism by which the probiotic bacteria help to improve the gut health? It competes. The first thing is the probiotic bacteria has a much better ability to adhere in the gut. And therefore, it competes with all other pathogenic bacteria in the gut and ensures that it stabilizes itself. So it secretes. I showed earlier that they produce a large number of metabolites, vitamins, other metabolites, small chain fatty acids, folate. All of these are very important for maintaining gut health. And one of the other things that they also are known to do is they present the antigens to the macrophages, and they help in phagocytosis. Now, what we also must understand is that we have already seen, they help to improve the tight junctions and prevent the leaking of the gut contents into the lumen. And they are also known to affect, or I would say, reduce the immune responses, which is constant when you have a gut-related disorder. So it helps to maintain the microbial balance, reduces bloating, diarrhea. It also helps to improve the levels of the happy hormones, as we call them, or happy neurotransmitters, the serotonin, GABA, dopamine, okay, reduces symptoms of depression and anxiety. Then in the immune system, it maintains the gut barrier, reduces inflammation, and helps to improve memory and cognitive function. So why we look at Bacillus amyloliquefaciens as a natural solution for leaky gut? We'll take a look at that. One of the things about Bacillus, unlike the other probiotic bacteria, is that it is a gram-positive spore-forming bacteria. Most of the others, the lactic acid bacteria, the streptococcus, the bifidobacteria, they are not spore-forming bacteria. Okay, so this is one of its advantages. The other thing is that because of their spore forming ability, they have considerable resilience in tolerating the acid and gut in the gut, the bile that is present in the gut, okay, unlike the other bacteria. Also, because of this heat tolerance, they also have an ability, if you're going to use it as a product, to have a greater shelf life, okay, along with their spore forming ability. What else do they produce? They produce various Cas-zymes, as we call them, proteases, amylases, cellulases. They help in digesting the food better. Okay, they produce a large number of antimicrobial peptides. Lactic acid bacteria by themselves produce a large number of bacteriocins, as we call them. Okay, antibiotics, they're also called. In addition, they also modulate the gut health. They are endospore forming. They prevent the biofilm formation of other pathogens because they have a greater adherence capacity than them. Okay. So we are going to take a look at this particular strain that we are touting as one of the best, basically because we have characterized it completely. We have looked at its genome sequence, analyzed its probiotic properties in silico as well as in the lab, and proved that this is a far better probiotic than many of the others. Now this Bacillus amyloliquefaciens was isolated from a deep sea shark, Centroselium fabrici. It was part of our Ministry of Home Science, Ministry of Earth Sciences project, okay, and it was isolated from 2000-meter depth. So it has a genome size of about 4.1 MB, around 4,200 coding sequences. This is the genome. It tells you about all the possibilities that are there the vitamins that it produces, the metabolic pathways that it carries out, the various secondary metabolites that it produces, and so on. It tells us about the various genes responsible for the probiotic property in the genome. It gives you information about the genes involved in acid and bile tolerance, vitamin synthesis, amino acid synthesis, addition, aggregation, resistance to antimicrobial toxins, environmental stress, sporulation, ion acquisition. But it also gives me information that this bacteria does not have any virulence genes in it, which is one of the most important requirements if we are going to use a bacteria as a probiotic bacteria. So the KEC pathway also gives you all the information related to the cellular processes, the information and all the other systems that are involved here. So if you take a look at this genome construction, you can see here that the outer circle of the genome actually tells us about the genes that are present, where they are present, and all of these which show the probiotic properties have been labeled in the outer circle. The enzymes that are present, those genes have also been found, and all of these aid in the digestion of the dietary fibers. This is one of the important requirements, remember, because when we talk about a probiotic, it also needs a prebiotic, because that prebiotic is the sustenance or the food for that probiotic. So it has to go together. So it has all these enzymes that are responsible for that. In addition, we talked about the antibacterial proteins that are present here. We modeled two of them. One is the LC1 and the other one is the amylocyclicin. These were modeled based on the enterosin and LC1 from Bacillus subtilis and found to have more or less similar properties. Now we have to ensure that this bacteria has the ability to pervade in the gut, so that it binds in the gut and stays there. So we used a very simple localization, labeled localization using fluorescein 5,6-FITC. Okay, and we found that it sits there pretty well even after 3 days after cessation of delivery of this probiotic. It is there for a considerable period of time. So how does Bacillus amyloliquefaciens support gut health? It restores the gut barrier, reduces inflammation. We proved this, okay? It lowers the inflammatory markers, TNF alpha and IL-6, enhances digestion because it produces the enzymes necessary, the Cas-zymes. It balances the microbiome because it inhibits the other harmful bacteria from acting and strengthens immunity. So it has robust survival. It has all antibiotic, antimicrobial properties. It produces surfactant, iturin. All of these we didn't model, but it's already present. It helps to restore the gut balance and produces enzymes. All the characteristics necessary for a probiotic bacteria have been proved for us in the lab to show that this is a promising upcoming probiotic bacteria. Now it is a versatile application. Remember, we isolated it from fish gut. So it can find application also in aquaculture. So it is suitable for human health, animal feed, for agricultural biocontrol. And because this is a grass organism, Bacillus amyloliquefaciens has been touted to be a grass organism. So it will definitely have benefits and it can be easily marketed as a product. My student has a startup marketing this as a probiotic product. So one of the things, it also reduces your support on the antibiotics as a necessary aid for treating inflammatory bowel disease. So that is about, we will not go on this because we look at probiotics. But we can say that we can give probiotic as a live bacteria to healthy individuals. But we have others who undergo treatment, like people who have HIV, people who are undergoing cancer treatment, or who have a immune system that is compromised. So for such individuals, you cannot give a live bacteria because this can itself cause a disease. So an alternative is to look at the non-viable probiotic, or what is called as a parabiotic. We have a project on this recently from ICMR to look at the impact of parabiotics or outer membrane vesicles of probiotic, to look at what they can do, because these parabiotic or OMVs actually carry a cargo, okay, which contains various enzymes, proteins, DNA, okay. So it has a whole lot of things which can find application. So we are now having a project working on this. So I have just put some comparisons to show how good it is. It is being studied even in the targeted drug delivery and for healthy gut therapeutics. So we are also having a similar project in our lab. So that is to compare. That's the last part of my presentation. I'm glad I could finish in time. This is Department of Biotechnology at Cochin University of Science and Technology. I would like to introduce this. We have a small group, people working on microbiology. Dr. Parvati and I. She works on marine ecology using metagenomics and microbiome, gut environmental microbiome. And other people who work on cancer biology, on brain research, Alzheimer's disease, other cancers, and so on. There is a plant group who work on conservation of mangroves and plant microbial interactions. This work was initiated when we did the metagenomic survey of the Arabian Sea and Bay of Bengal. These are some of the research projects that we did in our lab. Okay. Then we looked at the microbial communities of the mangrove systems, and I should say that we were able to compare the post-flood and pre-flood mangrove communities to find out the changes that happened in that. Because remember, in 2018, Kerala, the entire Kerala faced a massive flood or submerging. Okay, so we were able to take a look at that. We also looked at the community genomics of deep sea fish, when we looked for novel biomolecules and to look at diversity. There is a project from ICMR with Dr. Chakrapani, who works on brain diseases, to look at human gut microbiome and its implication in Alzheimer's disease. What I presented here was part of Dr. Venetia's work on genomic analysis of probiotic Bacillus amyloliquefaciens as anti-inflammatory disease. We looked at this in zebrafish model. These are the people who worked on various aspects of this genomics, gut genomics. The rest of them are sitting over there. I couldn't get a live picture from any of them, but they are all there here. Thank you so much. I'd be happy to have any questions.
I think very nice talk. I just wanted to ask you about FMT. What is your opinion? Because it has been shown, I mean, a lot of research has been done that they repopulate with healthy gut microbiome. Many drugs, like cancer drugs, they become more sensitive to the tumor. So what is your opinion?
I think that FMT is something that should be looked at with great consideration and hope, because in many times when individuals undergo therapy, chemotherapy particularly, there is no way that their gut microbiome can be established very easily. So the best alternative under such circumstances would be fecal matter transplant. But we must understand here that it is not just, there's a lot of processing that is involved. It is made clean and neat, and it's only the microbe that goes in there. The problem with FMT is that people view it with disgust first without looking at the logical implications of it. Because this really helps. Because when I talk about a probiotic bacteria, I'm only looking at the culturable component of the gut microbiome. I'm not looking at those unculturables which would in fact support the establishment of the or normalize the gut microbiome greater. So I really think that fecal matter transplant should be something that should be researched in considerable lot of time and effort should be spent on it to do so, because that is something. But we must understand that animals do it. We view it with disgust, but animals do it and we have seen it. So it is something that helps to re-establish the gut microbiota, which is extremely important. If you need to have a healthy life also helps to ensure that you're agile, you're healthy, you age slowly, all of those. You need a healthy gut. And if you need to help people who are undergoing various types of rigorous treatment, FMT is certainly a requirement. But we must understand here that FMT for these people would also come with a rider that they are already immunocompromised. Okay, their immune system would be hit very badly because of the treatment that they undergo. So it has to be done very carefully. So it cannot be a clinical trial alone. It has to be done under, how do you say, rigorous scrutiny by the medical fraternity. Because in disorders like Clostridium difficile, it is almost next to impossible to get the normal microbiome, because C. difficile is a considerably stubborn organ. It gets a very nice anaerobic environment in the gut. Happy to try.
Ma'am, thank you for your great presentation.
Thank you.
I just wanted to know, like, what are the food area, the nutritional part, how we can keep our gut healthy? Because I had gone through with the Ayurvedic treatment, in which you have to keep yourself eating for 2 days, and then you are like cleaning your gut, and to the some extent that it becomes a baby gut. They call it as a baby gut.
Yes.
And then they say that it is very sensitive and you have to take care a lot after that.
Yes.
So, and Yakult and all products are also there in the market.
Yes.
Like you drink.
Yeah.
So, and there are some products which you can make at home, like cucumber, radish, or some kind of like detox we use. So like how overall we can make ourselves like.
I won't go into the detox part of it or any of it, but I will look at the fermented foods that all of us consume.
Okay.
Fermented foods are part of the Indian food system that we have. We eat. I think Southeast Asia also has. I think Dr. would also tell us that there are large number of fermented foods. So fermented foods are something that we should make a part of our diet. I'm sure all of you would have had curd today after lunch, because that is something that would help to establish whatever is getting depleted. So I won't say anything about, because the Ayurvedic system will actually tell you that you have to re-establish your gut. Baby guts are extremely sensitive. We don't give babies burgers.
Yeah.
We don't give them fries and pizzas to eat, because we say that their gut is delicate. How on earth do you expect us to get back if we keep punishing our gut? वो हम भूल जाते हैं कि पेट भी अपना है, शरीर भी अपनी है. Disease हो जाए तो it is also our own. But you know, they were talking about the Pancha Indriyas in the morning. I was so amazed by it. This is exactly what it is, isn't it? Your nose takes you to the food. You know, and your taste perceptions are only limited to your mouth. के बाद आप उसको spit out कर दें, आप उसको वापस नहीं खा सकते, because it's changed in texture. You know, it's a visual, smell, everything, perception. That is what takes you.
It's how you control it. That is how you will be able to maintain your gut health. जरूरत से ज़्यादा भी नहीं खाना है, कम भी नहीं खाना है.
Thank you.
Thank you.
Can I have a quick questions? Because in our lab, we're trying to find the way to prevent calcium oxalate stone formation.
Yes.
So one of the ideas that's come across is we would like to find the probiotics that could use oxalate as an energy source or whatever,
Yes.
that can limit the absorption of oxalate to our gut. So my question is, do you have any idea how I can find this kind of microorganisms from fermented food or other thing, and how can I isolate the bacteria that can use oxalate?
You will have to modify your screening strategies because you're trying to look at oxalate degrading bacteria. So you need to look at that and probably devise strategies because you're looking at a culturable bacteria. So it's very easy to find. Yeah. So if you are saying that your source is fermented foods, so from fermented foods you can certainly isolate bacteria which would be able to degrade oxalates. Should not be a problem at all. Any other question?
Yeah. Yeah, thanks for the nice presentation, ma'am. You presented very well. So my question is about the FMT. Like sir asked about this and we're discussing about the same. Like for any disease, in some cases patient adapt the therapy, means respond for the therapy. In some cases patient didn't adapt or respond for the therapy, like in case of cancer and the chemotherapy and all these things, ma'am. Like is FMT a technique in which 100% acceptance by the patient is possible or not?
There are ethical, religious acceptance that must be there.
Ma'am, apart from the ethical and the religious view, means some good, healthy
Even FDA has only cleared it under certain conditions. Like phage therapy is only permitted under certain extreme conditions. When the pathogen cannot be killed by any antibiotic, they will say that phage ko try karke dekhte. So, FMT
one more thing, ma'am.
Yeah.
Like you have proven with your studies, what type of food we have to take and how looks like probiotics and all these are working fine to make a gut flora healthy and happy. So, ma'am, what techniques you have used for this to proven your experiments, like proven your theory, proven your hypothesis, ma'am?
This work that I showcased here about Bacillus amyloliquefaciens was please have a seat. Was something that was done by Dr. Venetia. That was part of her PhD doctoral research. She has completed all the wet lab experiments to prove that the bacteria can adhere, can form a biofilm, that it is non-toxic, non-virulent, and all of those things. In addition to that, we did a whole genome sequence analysis, which told us all the story about this bacteria, what it can do, what it can produce, whether it has any virulence genes, which can harm us anyway, because we are going to use it in very large numbers. So she did both the wet lab, which was supported by the whole genome sequence analysis. And that is how we proved that this is an excellent probiotic bacteria.
Thank you.
If there are no more questions, thank you so much.
Thank you, ma'am. Thank you for your enlightening lecture. Your insights on probiotics as the new building block of the gut health gave a new perspective on how microbiology is revolutionizing medicine. Thank you, ma'am. So we have our next lecture.
Our next lecture in the series is by Professor Dr. Dinesh Yadav sir. Dr. Dinesh Yadav sir is presently working as Professor and Head Department of Biotechnology, TDU, Gorakhpur University, Gorakhpur, India. Sir's area of specialization is molecular biology, bioinformatics, plant biotechnology, and enzyme technology. Sir published more than 100 research papers and many book chapters to his credit. Sir's dedication to education and leadership has been recognized with several prestigious awards, including Dr. Pushpendra Kumar Gupta Vishisht Krishi Vaigyanik Puraskar 2015 in the field of agricultural science by Uttar Pradesh Academy of Agricultural Science and Young Scientist Award. And today, Sir is going to talk on the topic Omics on intervention in finger millet crop for food and nutritional security. Let's warmly welcome our Professor Dr. Dinesh Yadav sir.
Thank you very much. So before I start, I would like to make one correction because my professor and head department of biotechnology is sitting as a chairperson. So I was an ex-head actually. This is one minor correction. Now regarding this, before I start my talk, I would like to congratulate the organizer and especially Dr. Sunil Kumar and his team who have managed very well, and they have given a platform for us also to share what sort of things we are doing. Especially I am specialized in plant biotechnology as well as we are working with the microbial enzyme technology. So today's lecture is basically focused on one of my area, and that is on finger millet crop. Everybody knows about this crop. It's a nutraceutical crop. There is a lot of efforts is being going on to enhance the yield of that particular crop. From a student point of view, this is very important in the fact that most of the students, now they are well aware about this particular topic, that is omics. The term omic is very much common nowadays, and it basically reflects the totality. When you study anything in totality, if you are studying the total number of genes of a particular organism, it becomes genomics. When you start studying the transcript, it becomes transcriptomics. And when you start trying to analyze the entire protein of a particular system, it becomes proteomics. And there goes on a lot of omics are coming. Now, the importance of this omic is that it basically reflects the advancement of the technology. And second important thing is that simultaneously you are able to analyze that out of this much number of genes, which particular gene might be associated with a particular function part. Now, my work is basically on transcription factor, and I would try to tell you that what sort of work is going on in the transcription factor. This is just the abstract part. For a student, this slide I always put, and this is some of the values which somehow we are losing. And this part I have taken from, if you remember the 10th or 12th, 11th, 12th class CBSE textbook, in the first page, they have mentioned these values. And this is very important. Always try to see this value. It may not be possible that each and every value you are trying to inculcate. But definitely, if you try to see this thing, some of these values will come into a picture and you should try to take them. Second slide, this is also compulsory slide for me. Why? Because we should always be pride of Indian scientists. And your aim should be there that you add your figure into this particular figure. That would be the major thing which most of the people used to think about it. And third slide, this is the outcome. You can say the person who are working with the science, they can achieve the highest award also. So these are three slides, which is very much important. I've already told you we are working in two different areas. One is on the transcription factor, and the other is on the microbial enzyme. In microbial enzymes, we are specialized with the pectinases group of enzymes. What is transcription factor for the student? Everybody knows this is basically a protein which regulates genes. And binding with the promoter sequences, you have a repertoire of transcription factors available. We are basically working with two different transcription factors. Why we are working with the transcription factor? This is also very important. Because as soon as the genome sequence came into the picture, or the plant genome sequences, people have realized that 5 to 10% of the total genome sequences, they are basically covering the transcription factors. They are basically responsible for it. Then they started working with the transcription factor. Then they found it is not just the one group of transcription factor. There are thousands of transcription factors available. So your basically aims that in which particular area, in which particular stress you're working, or in which particular area you're working, you may decide the transcription factor. Now, these are the two transcription factors we have worked. DOF transcription factor, this is a plant-specific transcription factor. You can see some of the characteristics we have put, that what are the major structural differences in the DOF transcription factor. And with this particular part, just working with the DOF transcription factor, and that too very simple approach, because you have a lot of limitations, but it doesn't mean that you cannot publish things. So we have worked with the bioinformatic aspect. The only thing is that we have increased the scale. We have worked on genome-wide identification of the DOF transcription factor in different plant system, and we found that this number of genes are there. And then we went to the expression part. So this all part, this story is basically related to the DOF transcription factor, and we got good publication, I can say, because these are reputed journals where we have published. Now coming to the next transcription factor where we would like to talk is, this is the nuclear factor gamma transcription factor. Nuclear factor, why we call it? This is a very unique transcription factor in the sense, this is ubiquitous. It is present in the animal system, plant system, everywhere it is present. And recently it has been reported in the plant system. This is a very complex, it is a trimeric complex. It has got 3 subunits. And these subunits, in the case of animal system, each subunit is being coded by 1 gene, but in plant, each subunit is coded by multiple genes. And they can combine also. So there is a lot of recombination in this transcription factor. These are some of the properties of this DOF transcription factor, that they are already present in all the living organisms. And this diversity basically depends on the genome size. So if you try to analyze the different genome sequences, you may have a different number of NFI genes. Today, I'm just going to present one of my PhD scholars. She is complete, Varsha, and her work, which has been done in the finger millet crops. If you try to see what was the major objective, there are 3 major objectives. I think there's some problem. The 3 objectives, I would say, the first objective was to analyze the finger millet genome and to tell that how many nuclear factor Y genes are there. Second was to validate this thing. What is the source of validation? You have to go for the wet lab. You have to go for the PCR-based amplification. You sequence it and you characterize it. And the third part, which is the most important thing, is that you have to see whether this particular genes might be expressing under some different types of stress. I would tell you one important thing. In agriculture, most of the time we are saying that we are trying to enhance the yield. But actually, it's not like that. It's not a question of enhancing the yield, because you know the area of arable land is limited. The only option which is left in the biotechnology is that the losses which is being incurred by biotic and abiotic stresses, that can be minimized. So if you are minimizing that loss, ultimately you think in terms of the yield increase. So we are always trying to see this particular transcription, whether it is related with some abiotic or biotic stresses. So these are some of the importance of the millet crop. Everybody knows it is already a harsh crop. It can tolerate a lot of abiotic stresses. But the problem with this is that the yield is very stagnant. And second important thing is, what are the factors which might be responsible for its tolerance? This is not well known. So one aspect is that the transcription factor might be one of the reasons which may be playing a very significant role. These are some of the types of millets which is very much popular in India. This is the crop which is nowadays people are knowing much about it, and its importance is realized. These are some of the data there. These are some of the nutraceutical properties. This is well documented. Everybody knows about the nutraceutical properties and why people are targeting. These are some of the values which has to be validated. Lots of things has been already validated. Finger millet genome, I told it was already available. These are some of the information about the genome. So this was our starting material from which we try to fish out that how many NFI genes are there. So these are the genes. These are some of the nuclear factor, transcription factor, which has been reported in other millets. As compared to the other millets, there has been no report in the finger millet crop. This is the first part. I have already told you the first part was totally bioinformatics. And this is the overview. You can see that how to predict that this much number of genes is present in their genome. So you need a specific domain. So every transcription factor has got a specified domain on the basis of which you can try to fish out that this much multiple genes should be there. So this entire approaches, we try to do it up to the structural analysis. We also find out that in silico, what are the diversity in the structural aspect of the transcription factors. These are the tools. I would just skip this thing. This is already available. Second part was the expression part. So here we have taken support from University of Hyderabad. We have collaborated with them because we don't have that particular facility, though that facility is very common. So we have tried to use this identified nuclear factor Y genes for the expression under the salt as well as in the drought condition, that how many genes they are overexpressing under these conditions. These are some of the stress markers, which is a very common marker everybody has to analyze when they are studying the transcription factors. These are the primers. These are the classical tools, expressive and profiling methods. I would come to the first part. So based on this identification, that how many genes are there, we found that there are 59 genes are there. So in finger millet, there are 59 nuclear factor Y genes. And how they are distributed, that also we have found, like nuclear factor Y, A is 18, B is 23, and C is 18. Rest, other bioinformatic aspect is that you have to find out whether that domain is present or not. So there is a lot of tools are there. This multiple sequence alignment will tell you that particular domain is present in that particular group or not. Because A has got a specific domain, B has got a specific domain. So based on that domain, you can try to identify that whatever you have predicted is confirmed based on that particular profiling. So these are some of the aspects. Then comes some of the in silico characterization. If you see on the top, there are various type of things are there. You can get the PI value also. You can get a lot of information. These informations are very important, but the only thing when it is validated in the wet lab, then only. But this gives a lot of information which we have collected, and based on this thing, we can see some of the diversity also. That in case of nuclear factor YA, what sort of diversity is there? In case of B, what is there? These are some of the things which I have already marked, that high molecular weight, low molecular weights are there. Similarly, in B also, you can see the same part. Similarly, if you try to see in the C genes, again, you have the same part. So you have collected a lot of information based on the tools. Then you can also find out the subcellular localizations or nuclear localization of these genes. Everything is bioinformatics. Till we are not doing any sort of wet lab based experiment, but this information may be important at the end also. So I would just go and tell you, these are some of the nuclear localization signals, which is prevalent in NFI, A, B, and C genes. Then these are the motif distributions. So we have tried to find out that what sort of motif distribution is there in the 3 different types of subunits. And this we have validated with the reported motifs, which has been shown in some other crop system. So these are some of the results in that way. You can see just highlighted this part. This is C. Then the final thing is that you have, since there is a lot of information available with Arabidopsis, rice and all that. So why not to what you have predicted, you can try to compare with the other systems and you make a phylogenetic trees. The purpose of phylogenetic tree is that at least something which has already been reported, and if that particular gene comes close to it, you may have a prediction that this might be also playing the same role. This is what the purpose of phylogenetic tree, when we have the information already available. So these are some of the, this is another additional things which we are doing related with the evolutionary significance of your genes. These are the gene structure analysis. You can also find out that how many introns are there. There are many genes which are intronless, but in most of these NFI genes, there is at least one introns are present in most of the genes. So this is some of the things which you can try to find out. Then chromosomal mapping can also be done by informatic based, that in which chromosome, how many genes are there. So we have many of the chromosome, multiple genes have been present. So this information can also be derived. Then comes the molecular structure. You can use this in silico based structural analysis. Then you can try to find out that whatever genes you have predicted, they can be subjected to the structural part. So then the structural means 2-dimensional and the 3-dimensional. And everybody knows that the sequence linearity, if you have the sequence, you can come up to the structural and the functional part. So that is one the basis of finding out the structure. These are some of the structural assessment. What are the criteria to find out whatever structures which you have predicted, this is correct, it is or not. These are the values which can tell you whether the prediction is good or not. These are some of the things like the residue in the most favored region is very important. It should be around 96% or 95%. So that is there in most of the structures which we have predicted. These are the modeling which we have done with the bioinformatics tools. I will not go in detail. There's a lot of information there. So you can see, based on just the bioinformatic tools on the predictions, we have tried to publish one paper, which gives you an idea that in finger millet genome, this much number of genes are there. And this much genes, they have a different characteristic using the various type of bioinformatics. So this is the first information, which is very important if you want to go for the expression part. Second important part is what is the structural and the functional diversity of this NFI transcription factor genes in the finger millets. So this is the overview. You can try to say that what sort of thing we are trying to make is that we are trying to make a docking study to reveal the functional attributes of the identified EC NFI genes of finger millet in stress signaling. And this is the chart you can try to see here, that how the NFI genes of finger millets, they are involved in the abscisic acid signaling pathway or not, we have to identify that part. Then for the identification of this thing, there is a specific receptor is there that is called PYL receptors. So if you want to find out this type of docking study, you have to know that how many PYL receptors are there. So we have tried to go further here. And we found that out of this, in finger millet, there are 14 PYL receptor proteins are there. So with this particular part, again, we try to find out the distribution of this PYL receptor genes, that where they are present, how many chromosomes, in which chromosome their distribution is there. So this type of data has been created by the bioinformatics tools. This is again the gene structure of the E. This is what we have done with the genes. Now we are trying to do with the receptor part. The same part is there. And the same phylogenetic analysis and the homology and modeling we have done. Some of the conserved region. The most interesting part is that when we try it, because this is a transcription factor. So ultimately you have to think in terms of the expression. So when we try to go for the cis regulatory element analysis of the promoter regions, you can see the percentage wise, the representation, that most of them, like 38%, they are hormone responsive. Light responsive is 25%. Then growth and development responsive is 29%, and stress responsive is just 8%. But what exactly we are thinking that in finger millet crop is already stable crop. And in that category also, there is only 8% of the genes which is responsive for the stresses. These are the structural assessment of the PYL genes. These are 3 genes which we have tried to identify the receptor proteins, and we have made a model, and then we try to use this with a finger millet genes, that is the NFI A and BC. These are the protein-protein interaction analysis. Again, using the bioinformatic tools, you can try to find out the protein-protein interactions also. These are the gene ontology. Again, if you want to find out the functional attributes, you can use this part. And based on this thing, you can see the 3 different functions, which is molecular, cellular, and biological process, and the involvement, which has been highlighted here. So that could be a very significant role which this particular transcription factor will display. These are the ligand interaction. You can try to find out the active sites also. The active sites of the proteins, which might be playing a significant role there. So this is the way of interactions and ligand studies. These are some of the reports of each proteins. This is the protein structural modeling. Then these are the ligand structures which we have tried to go for the docking studies. These are the protein ligand interaction. You can see these 3 proteins, and we have gone for the different types of interactions there. This is the stability analysis. These are all tools. These are all available tools. The only thing is that you have to think about the target and the receptors and try to interact with them and get the outcome of this thing. So to make this story very short in that, this is the outcome of this thing, is that the PYL might be interacting because the promoter region of the PYL receptors consists of CC80, and CC80 is the very common promoter sequence in the NFI genes. So therefore, to examine the interaction, whether it is happening at the promoter or the coding region of the PYL genes, we perform docking between the ABA receptor and the NFI transcription factor of the finger millets. So these are the protein interaction. In order to find out, to validate this thing, we have done this protein interaction, protein-protein docking experiments. And these are some of the results which you can see here, that how they are interacting. And this is the shaded portion. You can see some of the highlighted residues. These highlighted residues may be very significant when they are trying to interact with the receptor proteins. So based on that part, the another thing, again, it is a bioinformatics, nothing but lab here. So we got another publication which is in crop design, where we have tried to go for the molecular docking insights into the NFI transcription factor and the PYL receptor protein, reveals that abiotic regulation is there in finger millets. Now comes the last part, which was the expression part. And this is very important. And the only thing when we talk about expression is nowadays, it is the real-time based expression studies. So you need a real-time piece here there. So we have collaborated, I have already told, with the University of Hyderabad. So we have used 2 different varieties, and these varieties were sensitive and they like drought and the salt sensitive and the resistant varieties. And these varieties were subjected to the different types of stresses. These are some of the conditions which we have given for the stress condition, which is a very common. When you talk about salt, you had used a particular concentration of the salt. So this type of things we have done, and we have used different time intervals for different intervals, 6 hours, 12 hours. These are some of the results. These are some of the parameters. I have already told there are some stress markers are there. These are the stress markers, like proline content, you have to check and how exactly it is changing. Similarly, there is a membrane stability index that is also reveals the ability of the abiotic stress tolerance. Then comes the MDA content. So based on this thing, you will see what we have tried to find out. These are some of the, this figure will tell you the entire story. If you try to see this figure, the central part is something which is very important, where these are the main genes which might be associated with the different types of the hepatic stress. It could be 1 gene, it could be 3 genes, it could be 4 genes. So this type of comprehensive expression analysis will ultimately come that this much number of genes might play a very significant role for the expression. So based on your down regulation, up regulation, you try to predict it, which can only be predicted when you go for the real-time based expression analysis. So these are some of the shaded things, which is very important here. You can see same thing presented in a different mode. Each individual genes and different hours, you can see. Some of the information which I have told you, these are the 3 genes which is very important as far as the tolerance is concerned. This is in case of salinity stress, again you can see 16, CY, C2 and B10. These are the 4 important genes when we talk about the salinity stress tolerance. This is the again phylogenetic tree based on the stress tolerance, that these are the important genes which may be playing a significant role. So finally, to conclude, what we have done is that we started with mining the genome of the finger millet, and the purpose was to mine that how many nuclear factor Y genes are there. Second important is whatever you have mined, you have to validate it. The very simple thing is you go for the PCR based amplification, sequence it and analyze it. The third part was that under salt and drought tolerance, which of this NFY genes might be playing a very significant role. So we came to the conclusion there are 3 to 4, 5 genes which might be very important when we talk about the salt or the drought tolerance. So this last part was expression. So we published this in Planta Journal. So each story we made in such a way that it ultimately outcome should be one publication. Very simple work, but the only thing is that it is a very systematic work. If you try to do this thing, it can come ultimately end up to this part. Another thing we have done is that this is one of the copyright we just received, and this is related with the overall strategy, what could be done in the finger millet crop, targeting the nuclear factor Y. So this is the copyright which has been registered recently. And this is the ultimate person who are basically working. I'm just coming here and I'm talking, but these are the entire students, 16 scholars, they have already completed PhD, 5 are registered, 5 postdoctoral students they have completed in my lab. And one of that postdoctoral is my wife itself, Dr. Sangeeta Yadav, and she has initiated the work of pectin lyase. In fact, she was the first person who did MSc, PhD, and last 9 years project in DST, exclusively working with the pectin lyase. I think most of the people, if you just put pectin lyase, Dr. Sangeeta Yadav's name will come because she has done a lot of work. I have added the molecular part and the bioinformatics work. She is an enzymologist. She did all the enzyme part. I have just added the molecular and the bioinformatics. So thank you very much. If you have any questions, I would be happy to answer it.
No questions. Any outcome of your studies has been translated to the field to increase the quality or the output of millet?
So as far as agriculture part is concerned, so directly it is not like that. But as far as you can say the enzyme part, we have got 7 patents we have filed that has been published. That is mainly related with the application in the rating of the fruit juices, rating of the natural fibers and fruit juice clarification. That part is very good work. Because in plant system, it is very difficult to translate what sort of transcription factor work which we are doing directly into the field part. But what we can say that we have identified, for example, 5 transcription factor genes. If you associate with some plant breeders and this part, we can try to go for further studies part. But as such, I will not say that it will directly come to the field part. So that is the outcome. Yeah, definitely. So that is there.
Yeah, Dinesh, very good presentation.
Thank you.
Very clean data, bioinformatics data.
Yeah.
I was just wondering, NFY is a transcription factor.
Yeah.
So among the class of transcription factors, is it the zinc finger or leucine?
No, zinc finger is a different, sir.
Right.
This is not a zinc.
So how does it bind to DNA? I mean, if it is a transcription factor, it must bind to DNA, or one of the transcription factors.
Yeah,
auxiliary.
actually, sir, what happens that this particular transcription factor, it binds to the CCAT promoter sequence.
Right.
Yeah.
So it binds to it with some motive.
Yeah, definitely.
So is there a consensus sequence of that motive?
Because in this, there are 3 subunits. First subunit, which is present in the cytoplasmic region, its other subunits comes into the nuclear. They all combine together and then they interact with the promoter sequence, that is CCAT.
But what I was trying to say that if you have DNA fingerprinting experiment, let the transcription factor bind with the DNA, cut with DNA is one, remove the transcription factor, see what is the consensus sequence.
Yeah, that's true.
It may be cysteine rich or leucine rich or whatever.
Yeah,
Thank you so much.
Thank you, sir, for your remarkable presentation. Your insight into finger millet.
I think just to add on what sir was trying to say is like now the DNA binding binding site selection assay. So
it is there because whenever you talk about a transcription factor, there should be a DNA binding domain.
Exactly.
There should be that is there.
No, I mean even if you have a transcription factor where the binding is not known, you can generate. You can just do like now binding, pull down with transcription factors.
Yeah, sure.
Cut the sequence and sequences like now, and we can find what is the sequence identified.
Sure. Thank you.
Thank
you.
Thank you, sir, for your remarkable presentation. Your insights into finger millet and its potential role in global food security highlight how scientific advancement can address critical challenges in nutrition and agriculture. Now, we have our last speaker for the session, Dr. Manvendra Singh, sir. Sir is an associate professor in the Signature Research Program in Cardiovascular and Metabolic Disorders at the Duke NUS Medical School. He also holds an academic appointment in the National Heart Research Institute, Singapore. At the National Health Center, Sir specialization in the developmental biology, cardiovascular biology. Sir is currently affiliated in the Duke NUS as a faculty member and was awarded as the NRF Fellowship to pursue cardiovascular research. Sir's long-standing research interested are in the areas of congenital and adult cardiovascular disease. And today, Sir is before us to present the talk for his lecture entitled Emerging Roles of Hippo Signaling in Heart Failure and Therapeutic Opportunities. Let's warmly welcome Sir Dr. Manvendra Singh, sir.
So good afternoon, everyone. I know it's very difficult to stay awake after lunch, so I will try to keep it brief and try to not to show too much data, but give you the message what we are trying to do at Duke NUS Medical School. Before I start, I would like to thank all the organizers and volunteers for putting this program very nicely and very well organized. So by training, I'm a developmental biologist, and what we are trying to do is trying to use our developmental biology expertise to understand cardiovascular diseases. So if you look at the data, no matter where, whether it's in India, US, Europe, 30% of all the deaths are because of cardiovascular complications. Here you can see cardiovascular prevalence within India, you can see is significantly rising. This is from, still I think is from 2020. If you look at before, it was much lower. Now it's increasing. Amongst the cardiovascular diseases, if you look at the different type of cardiovascular diseases, the ischemic heart disease is the primary complications. What happened in this case is when there is a blockage of the coronary artery leading to hypoxic condition. So the cardiomyocytes or the heart cells, they don't get enough oxygen or nutrients. So majority of the cells, they die and they create a vacuum which is filled by other cells which are non-cardiomyocytes like fibroblasts. And these numbers are going to significantly increase because of two major factors. So the one which we discussed briefly in the morning, whether aging is a risk factor or aging is a disease. There is a significant correlation between age and cardiovascular complications independent of the age and gender. And these numbers are going to increase because of global aging. We keep saying that India is a young country. Okay, we are young compared to maybe China, but still we are aging population. There are going to be more number of people above 65 in the coming decade. So we got to be prepared for that. This we can try to delay, we cannot prevent it. As I said, aging we cannot stop. All of us are going to age. Another factor which we can prevent, which is relevant to this conference, is this aspect. So risk factor. What we are doing is we are facing a life-threatening lifestyle challenges. This is what we are becoming. Look into any newspaper. India, international, everywhere, this is what happening. Obesity is the major problem. We are eating. The major risk factor, if you look here, diet. Can you believe what you are eating is the primary risk factor for ischemic heart disease? And this is like a statistic. See, we all know, we all talk about it, but I'm trying to show you the data, which is published data. Look here, dietary risk is the first, 70% associated with ischemic heart disease. And not only dietary risk, if you look at everything else here except air pollution, is all associated with diet as well. Like whether it's a high blood pressure, cholesterol, tobacco use, glucose. So everything, all your diet is affecting your heart. So you are primarily responsible if you have a cardiac arrest. You cannot blame anybody else. So we need to do something about it. And what we can do, but before I say, one more point I want to make it here is the air pollution. That's another risk factor for cardiac diseases, but not only for cardiac, it is also for lung disease. So you can see this is the second most prevalent disease in India after heart, COPDs. And you can see that how this map or the prevalence has changed from 1990 to 2016 because of all these risk factors for COPD. And these two diseases are the major problem for us going forward. So if we, in real sense, if we want to fight against cardiovascular complications, we have to not only focus on disease center approach, but we also have to look into preventive measures, what we can take. And that is lifestyle changes, or what we call Ayurveda. It's same thing, it's just a different name, it's a more fancy name. If I say lifestyle medicine, then Ayurveda, but it's exactly the same thing. So let me talk about a little bit about the disease center approach, what we are doing in the lab. And then I will tell a little bit about what we can do when we talk about the prevention, what you can do to take care of your health. So my lab studies heart. So as I said, I'm a developmental biologist. I look into heart development, look into congenital heart defects. So this is the structure, what is known as a proepicardium. This is the heart. This is the fetal heart, developing heart. So at this stage, the heart has only two layers, endocardium and myocardium. And these cells, these proepicardial cells, during development, what they do is they proliferate, expand, migrate over the heart, and within like 2 to 3 days, they form the outermost layer of the heart known as epicardium. Soon after these cells they have covered the heart, a subset of these cells that undergo epithelial to mesenchymal transition, they invade the underlying myocardium and they become fibroblast endothelial cells or smooth muscle cells. And these are the cells which make the coronary vasculature. In addition to that, the epicardium is also a signaling center. So you can see the normal heart, if you take a section of the heart, this is how it looks. So you have an outer epicardium, then you have a compact layer myocardium, and then you have a trabeculated myocardium. But if you generate a heart where there is no epicardium, this is how it looks. So this heart cannot function because there is no thickness. So if there is no thickness, heart cannot pump blood. So that's where so not only the epicardium act as a cell source, but also act as a signaling center. So in addition, in adult, so during development, most of these signaling pathways we talk about, they are all active. But in adult setting, they are all shut down because there is no need for cell to proliferate. They are terminally differentiated. But what happens to the epicardium is when there is a ischemic injury, there is a coronary blockage, the fetal gene program is activated in the epicardium. So you can see here, shown here, this is the heart. You can see this red cells suggesting that compared to uninjured, the injured heart has activation. And then these cells, they start proliferating, and then they start expanding and differentiating into cardiac fibroblast. This is like an experiment. This is called a lineage tracing experiment. So this is like, you can tell a fate. It's like astrology, right? Somebody can tell what you are going to become. So this is an experiment you do in a mouse model where you can label these cells. So this is the uninjured heart you are looking at. And you see these blue cells only here, only one thin layer of blue cells you can see. But if you look at 2 days, 5 days, or 7 days post ischemic injury, means post heart attack, you can see there's a lot more blue cells here. And this is just to show the majority of these cells are fibroblast. So when you have ischemic heart problems, cardiomyocytes die, and the heart is repopulated with these cells, which does not have the beating capability. And what then happens, your heart is not going to beat properly, and you will have arrhythmia. Means you have an irregular heartbeat, heart function, and eventually you are going to collapse. Epicardial cells also secrete a lot of paracrine factors. So shown here from the adult heart. And these paracrine factors, what they do is they try to help your heart to repair. So see, the best response is a nature's response, right? The heart has evolved for so long. So the immediate repair, the regenerative or reparative response you get, that's the best response you can get. That's the best treatment you can get. And epicardium has a significant role to play in that aspect. So these are the reason why we started looking into epicardium. This is the signaling pathway, which I'm going to briefly talk about and show you why this is relevant to the condition I'm talking about. So Hippo signaling pathway, this was identified initially in Drosophila. Majority of the core components are very conserved, like from flies to mammals. And they were identified as a regulator of organ size. So so this is like a kinase cascade. You can see MST1 and 2, LETS1 and 2, YAP-Cas, which are transcriptional coactivators. So when they are phosphorylated, they can stay in the cytoplasm. Or when they are not phosphorylated, they can translocate to the nucleus, interact with the transcription factors, regulate target gene expression. So when I mentioned that they are implicated in organ size control, is that if you inactivate, suppose in a mouse, if you delete this gene, your heart is much bigger than normal. Bigger doesn't mean it's much more functional. It is just bigger because the cells, they proliferate more. The reason I got interested in this pathway because YAP-Cas. So we decided to look at this signaling pathway by manipulating YAP and TAS in the mouse heart. So if you look at the expression of YAP, I'm not showing all the data, just like now trying to make the point that the majority of the Hippo signaling components are expressed in these cells. Not only in the epicardium, which is you can see here, but also in the myocardium. So at this stage, when there were already few people in the field working in United States, and what they made the point that YAP regulate cardiomyocyte proliferation. So we asked the same question, during development, what role they have to play in the epicardium. As I said, epicardium can be a cell source for coronary vasculature, can be also helpful for the thickness of the myocardium. So we found that is expressed. So we did like a genetic experiment. We got the flox allele. So what you can do is you can literally delete YAP only in these cells. You can see the green cells. You can do tissue-specific deletion of these transcription factors, transcription co-activator. And that's exactly what we did here. We use a Cree-LoxP system, so Cree mice crossed to a flox mice, and we generated a knockout mice. So if you look at the mutant mice, or the if I say YAP has knockout mice, double knockout mice, you can see the heart has a severe hemorrhage. If you look at the heart, is not formed properly. But that was not our interest. Our interest was looking into coronary vasculature. So I decided to generate an inducible. So what the beauty of this system is, you can have a inducible Cre line, which you can activate by giving a drug. So if you give a tamoxifen at a certain time points, you can activate the deletion at that time point. So you control when they are activated, when they are deleted. And what we found that the coronary vasculature were severely affected. And then what could be the reason behind that? So we look at the cell proliferation, we look at epithelial to mesenchymal transition, and we also look at the cell fate, what the fate of these mutant epicardial cells can become. And what we found that in absence of YAP-TAZ, the epicardial cell proliferation was impacted, the EMT was impacted, as well as their cell fate specification was impacted. And this is what we eventually concluded, that you need this YAP-TAZ to regulate some of these expression of these transcription factors to regulate epicardial cell proliferation, EMT, and cell fate. So this was the developmental study, which, well, as I said, I'm a developmental biologist, so this was interesting for me. But then I ask a simple question, right? In this system, if we are deleting YAP and TAZ, you are reducing the epicardial contribution to fibroblast. If we do the same thing in an adult setting, that could be very interesting, very, very significant, because you can reduce fibrosis, you can let cardiomyocytes to proliferate, and that can heal the heart faster. So we did. So before I explain, so when you have a ischemic injury, there are three stages. Initial stage, like any injury, you have an inflammatory response. Then you have a proliferative response where the fibroblasts, they proliferate. And the third is remodeling, when you have excessive collagen deposition. So we generated, again, inducible mice, and we deleted in the adult heart. So what we found that loss of epicardial YAP-TAZ result in severe cardiac defects, severe cardiac problem. Majority of the animals, they died. Instead of living longer, which we expected, they all died within 2 to 3 weeks of age after having a ischemic injury. And when we look at the heart, we were hoping to see less reduction in fibrosis, right? Based on our embryonic study. But if you look here, so the blue cells, you see they are fibroblasts, and we see massive increase in number of fibroblasts. So this was completely opposite of what we were hoping to achieve with this deletion. So how to explain when you have completely opposite result? What we noticed that when we open these mice, their chest cavity, we see a severe inflammation. So we thought maybe inflammation has a role to play. And then we perform an immune array on the ventricles. And to cut this story short, I'll say we identify a bunch of cytokines which are secreted from the epicardium. Shown here, one of the candidate was IFN gamma, is produced by activated epicardial after injury. And we showed by using chromatin immunoprecipitation, promoter analysis, that YAP-TAZ can directly bind the promoter of IFN gamma and regulate its expression in the epicardium. One of the established function of IFN gamma is to regulate homing of regulatory T cells, which have anti-fibrotic effect. So what you can see in the injured, 7 days post-MI injury or 14 days, in the knockouts, you have significant reduction of the T cells. So there is a less T cells which have anti-fibrotic effect, means there is a more fibrosis. So that's one explanation what we came up with. Then we tried to prove the point that IFN gamma has a role. So what we did, we treated hydrogels with IFN gamma. So this is the empty hydrogel. You can see the blue staining as a fibrotic tissue. And when you put the IFN gamma hydrogel treatment, you can see the fibrosis is significantly reduced. And this has been shown by other people that if you can deplete, you can delete the T-rex cells, you can minimize their number, you have the similar effect what we see. So our results was very consistent with the known literature. One thing which we did not
go in detail in this JCI paper was that we also found that there is an increased homing of macrophages. And macrophages, what they have been shown to play a very essential role in angiogenesis during development, but in adult heart, they regulate fibrosis. So this is just to summarize what we have found in this epicardial mutant, that instead of having a reduction in fibrosis, we see increase in fibrosis. And what we believe that this is because of the overactivation of the immune response. So what is happening in fibroblast doesn't matter if there is excessive inflammation, it can override the fibrotic response. So how to prove? So that's the hypothesis, how to prove that. Here we are deleting in the epicardium, which is affecting the fibroblast, affecting the immune response. So what we can do? Let's delete only in the fibroblast or only in the immune response and see what we get. And this is what we did in a completely new study. So the study has been already published, so you can read if you are interested, but I'll just try to summarize what we have found. When we delete now only in fibroblast, only this epicardial derived fibroblast without touching the inflammatory response, now we see there is a significant decrease in fibrosis and there is an improvement in cardiac function. And this is what we were hoping at that time. We also found that YAPTAs are activated in macrophages. I think in okay. So there are two different macrophages, M1 and M2, depending on their polarization, and pro-inflammatory M1 macrophages. The polarization status of these macrophages determine how the fibroblasts are going to transform into myofibroblast. And these are the bad cardiac fibroblast or myofibroblast. These are the fibroblasts that secrete collagen and make the heart stiffer. So the heart is not able to beat. I will just try to summarize what we have found in this study. Again, is published. What we found that the YAPTAs, what they do is they balance the number of or the behavior of M1 versus M2 macrophages. So they can directly bind to the IL-6 promoter, regulate pro-inflammatory expression, or they can act as a repressor in M2 macrophages by interacting with HDAC3 and co-repressor complex. When you delete YAP and TAS, there is a reduction in pro-inflammatory, so you reduce the inflammatory angle, but then you elevate the pro-fibrotic or anti-fibrotic macrophages, and that seems to help with the reduction in the fibrosis and increase angiogenesis and improve the cardiac function overall. So both of all these three studies, what they suggested, that we can target YAP and TAS, and if you target in these two cell types, that can help to improve cardiac function. So this is just summary. I'll not go into too much detail, but cardiac fibroblast, you have, again, in fibroblast, they are pro-fibrotic, pro-inflammatory, same thing in macrophages. So if we inhibit, we are going to reduce the adverse effect and improve cardiac function. And so we asked another question, whether this is only true in heart, because immune cells are everywhere in body, right? Wherever there is an injury, there are immune cells. So we asked what happens if we delete YAPTAs in immune cells or macrophages, and what happens if you generate a lung fibrosis model? And what we found that if you delete YAP and TAS, you can reduce lung fibrosis. Most of the mechanisms shown here, so I will not go into too much detail. But what we found that, is not a heart-specific response. You can use in any fibrotic tissue, whether it's lung or heart. So what we are doing now is, what we are doing is now we are trying to take a step further, see whether we can pharmacologically target YAPTAs. So we have collaborated with these two companies in the United States, Genentech and Vivace. Vivace is already, they have developed an inhibitor which can inhibit YAP activity. They are testing, they are already doing a stage 1 clinical trial for a cancer. They are not interested in heart so far. So we are going to get those drugs. We are going to test in our ischemic heart model as well as in pulmonary fibrosis model. So that's the stage where we are with this, the YAP and TAS. Coming back to this is not working. Can you check? Okay. Okay. Coming back to what we can do in terms of prevention. So before I go in detail, this is like a disclosure I have. I have a co-founded a company called Nova Health Nutrition, and this is to promote healthy aging. So coming back to the risk factor, as I said, dietary risk factors are the major problem. And this is the statistics about the obesity challenge we are facing as a country. By 2050, like we have like around 500 million people. So that's like roughly 1/3 of our current population are obese. And obesity is going to affect everything. As I said, like no, whether it's a cardiac complication, any other disease what we have can be associated with obesity. Okay. So if you look at this chart, so this is the reality of today. If you look at this chart, majority, as I see here, like 50% of the patient, when they are around 45 to 50 age, they have at least 4 chronic conditions. Not 1, 2, at least 4 chronic conditions. So if you are in 40 to 50 range, just be aware. In a country like that, like India, if you look at the healthcare costs, it can significantly increase over time. And we are the country where mostly all the expenses come from your own pocket. So don't do it for government, don't do it for anybody, do it for yourself because it's your health and it's your money. So work for yourself. So if you look at the functional capacity, so this is how your life should look like. The black line. If you have a congenital condition, you cannot do much. You already have a limited functional decline. These yellow and orange and green line, this is the lifestyle challenge, the lifestyle modification you have created in your life. If you do it in early stage, when you are a child, you are obese child, this is the trajectory you are going to have. If you have made mistakes in your adult life, you are eating everyday McDonald's fries, like no burger and fries, and drinking Coke. This is where you are going to go. So you need to fix your lifestyle. So the idea of lifestyle medicine is this. So if suppose you have a healthy 50 year of lifestyle, and then you are slowly start getting a problem, you have another 30 years to live, you can live in misery or you can try to do something about it. What you can do, of course, most likely you will not be able to change your lifespan. Even if we say like, okay, you are going to live the same age, you can live in a good life or bad life. This is what you should be targeting. From 50 to 70, can you make a shift in this direction by changing your lifestyle or following Ayurveda? You can. And I'll show you some data which is very relevant by doing very simple thing in your life. Not complicated diet you have to follow. Let me show you the data. It's called calorie restriction. This is like a paper published by Fontana long time ago. There are numerous papers you can find. Calorie restrictions without malnutrition increase health span. So this is normal lifestyle in a mouse. This is normal mouse. This is the obese mouse. When you cut down the calories, you can shown here 85, 50, 40. You can see the life extension of these animals. So it's shown in mouse, it works. What about monkeys? See, this is the normal aging mouse. This is with calorie restriction. You can see the age-related mortality is reduced. Okay, whether it works in human, I mean, that's most relevant for us, right? Yes, it does work. This is the paper from Fontana, again, published in Science long time ago. I had a privilege to meet him before coming for this trip. He was in Singapore. And you can just see, this is the same person. It's not even different person. It's just this person presented here. His body weight was 81. He cut down to 60 kg almost. You look at the parameters, cholesterol down, LDL down, glucose levels again down, blood pressure all in normal range. It's like resetting your body by just doing one thing, just cutting the calories, right? Even if you look at long-term, the cardiometabolic and vascular effect, you can see, look at all the parameters. And this is by just a simple trick of cutting down your calories. And this is not only true for cardiovascular complication. See, look at this. The moment you reduce the body fat, you have a reduce cancer risk, cardiometabolic risk factors are down, pulmonary disease, everything is like you are resetting your body. So what I'm going to say is like, just do this experiment I did on myself during COVID. I was like 92 kg. I cut down to 75 and I could see my whole blood tests shifting. So just try, is a very simple thing. Whatever you are eating, just eat. Okay, just count the calorie you are eating. There are apps, free apps, mobile apps. You can count like now if you are eating paratha, how much calorie roughly it has. Make a list and just cut down total number of calories. Not like now shifting from four paratha to 2 paratha and then eating like 5 gulab jamun. It's not going to change. You have to count total calorie. Just change little bit, reduce 20% to 30%. Trust me, you are going to fix most of your problems. Okay, so this is just summary. I think I'm about to finish. So as I said, like no, do this. It has been proven in a mouse model, monkey model, human model. I don't think so. And I have done myself based on this. So we always talk about this prevention is better than cure, and then we go home and we eat and then we sleep. We don't follow anything. Okay, it's time to make some changes in your daily routine. Okay, because what you don't want is to become this. You don't want to become a subject, right? I was in IIT Madras before coming to Gorakhpur, and there was a conference on cardiology, and people were discussing, okay, the subject this, and then had this problem. This is what you don't want to become, right? So do a simple thing. Take ownership of your health, okay? No one else has a more stake in your health than you. So with this message, I would like to just acknowledge my team, my lab members, my funding agencies, and my disclosure. And I'm happy to take any questions you may have. Thank you for your attention. Yes, please.
Very nice presentation. And I would like to know, like you have depicted that the data shows that men are at higher risk when dietary restrictions are not being followed more by the men, because it is one of the major reason for the cardiac diseases. So can you like say or identify like what are the factors why dietary restrictions are not, like they are not able to restrict themselves? What are the conditions, like what are the factors responsible, and so that we can have some kind of comparison that why women are not so risk. Yeah.
So it's not true that women are not at risk. They are equally at risk. So if there are men and women healthy, of course men has a more they are more susceptible, and there are various reasons about that. Okay. You can say traditionally, men were supposed to build the country. You were supposed to take care of home. That's one thing. You have different hormones than a man has. That's another issue. There are genetic factors. There are like no social as well as genetic factors behind that. But if you take both people and then put them on diet, similar high-fat diet like now McDonald's food, you have equally you are at risk. There is no benefit of being a woman at that now in that context. Yeah. So try not to blame your husband or you know your male partner, but like try to stick if you can.
Sir, very nice presentation and it was very connecting. I would like to know one very important thing, like diet is important, but like sometimes we read in the newspaper that almonds are good. Suddenly we know that chia seeds are good. So whatever we read, we buy. And then we don't know how to eat, like how much quantity we have to eat and all that. But when it is connecting to our heart, so what are the dietary or like you can suggest that we should eat and take care of our and also the timing is very important. What I have observed in my case, like when I used to do this Vipassana, you heard of? No.
No.
Vipassana is a meditation center where there are some timings. So when you, it's not intermittent fasting, it's a timing process, like how we eat. Like 4 o'clock or 7 o'clock we eat, and by 11:30 we stop eating solid foods. We can have liquids. So in that terms, you find your body in a very best condition because it's repairing and all that state. So how we can connect this to our heart factor?
Yes. So the primary, as I said, like no, that's why I in the morning when the Tripathi sir like no, he said, I said like no, I told him, if I tell you 100 things to follow, you are not going to be able to follow.
Yeah.
And this is like a completely wrong mindset. If I say almond is good, then you buy almond. Then tomorrow I'll say apple is good, then you start using apple. So the solution, what I think is just whatever you are eating, whatever daily routine you have at the moment, the first thing you do, just cut down the calories. Okay. Just go get your blood test done, regular like no checkup, nothing complicated, very simple like no like 500 like $1,000. You get your things done, checkup, and you just do the calorie restrictions what I'm asking you to do. And then you get after 3 months, 4 months, 6 months, you just do the same test again. And just look at yourself, like just compare the chart. I have done, that's what I'm saying. I did it. See, this is not going to cost you anything.
Yeah, but this B12, D, all deficiency we come to know.
So the first step is this, right? This is first step. Once you get back to your normal life, you get back to your normal body weight, your BMI is normal, then the second step comes.
Okay.
That is nutritional deficiency.
Okay.
Majority of the Indians are deficient in iron, vitamin D.
Yes.
Can you believe living in India and you are vitamin D deficient?
Yes.
Like we have sun outside, right? If you go, but most of the time people are working inside buildings.
Yes.
You're not exposed. You're not working in the field, right? So major population, everybody wants to have an office life, right? And that's what they have. That's why you study. That's why you do what you do, to have that lifestyle. But that has a complication. So the first step, fix your BMI. The second step, look at what deficiencies you have. You can start taking, if you have a vitamin D, just take a multivitamin for 3 months, like you are fine. You don't need to think about D, E, F, G, H, I, J. No need. So just 2 things you have to do: calorie restriction and find the nutritional gap what you may have. If you have an iron deficiency, take an iron capsule. Because you are not going to be able to change. If I tell you 20 vegetables, so somebody in the cardiology conference suggested that we are supposed to eat 6 servings of vegetable. I said like, no, sir, maybe you are a professor at like IIT Madras. Tell me how many serving you had today. Nobody does that. You cannot. It's not possible.
Correct.
Simply is not possible.
Not possible.
What the reason I'm putting so much emphasis on this is because this is doable.
Yes.
I'm not asking you to change anything. Just cut down the quantity,
BMI and quantity.
but the total quantity, not chapati and then replace with something else.
Yes.
No, total number of calories you cut down. I can bet anything on that you are going to see the change in your reports without doing anything. Then you work on the nutritional deficiency, which you can take nutraceuticals. There are tons of companies selling nutraceuticals, like Ayurveda products you can take.
Yes.
That can put like most of the vitamins, nutrients, like micronutrients, everything you can supplement.
Yeah. Thank you.
Yeah, please.
It seems to be really interesting. This is the kind of a concept among all of us, especially me. Is there any estrogen protection for women towards the cardiac diseases?
Yes, yes. As I said, like, no, if you are a healthy woman and if you compare healthy men, healthy women,
Yeah.
then there is some benefit because your heart is smaller than your partner, your men heart, right?
Okay.
You have a different body composition than they have a different lifestyle compared to the. So there are differences, and you are an advantage on that, like everything else.
Yes. Other thing is like, though we said there is no early onset, specifically this estrogen protection for the women, but nowadays I think in Sri Lanka we can see that early onset for the CAR disease. That means like it's diluting the estrogen protection for us.
Yeah, yeah, I think, in fact, I was curious, Manavan, very good presentation. I was curious to tell that estrogen is a protector. Estrogen has a cardioprotective role. You have advantage. So that part is very clear. But now problem is that I had told that every society, women live longer than men because of the reasons. Now things are changing.
Yes.
Like you and I are subjected to the same stress. Yes. You work, I work, you have your boss, I have my boss, so we have issues. So that gap of 5 years or 6 years between men and women is going to narrow down. But definitely women on all cardiovascular issues, they do have little protection against the men. So it's established. And coming to your lecture, I was very happy because this topic has been close to my chest for almost 30 years. And you know the figure which he shown that monkeys, because he had to cover a lot of data, that figure was published in Science, and I took it, I think, several years ago. The importance was that look at the front view and look at the side view. And you'll find the difference in both the dietary restricted, and it's a longitudinal study of 26.5 years, not 1 year, 2 years. They had a longitudinal study. At the end of the study, they had taken these photographs. The calorie restricted one, the dietary restricted, whatever you call it, CR or DR, they look much healthier and not that frail as the control one. So excellent study. The only thing, Manvind, I would like to suggest you, change that photograph with your picture with 95 to 75 and the parameters. If you have the data, that will be wonderful.
Actually, I have a picture. I can show you, but it is without a shirt.
Because,
So actually, I was thinking to replace that, and I had like, no, I can literally pull my blood.
But it's good, but don't show it in India, at least abroad.
So the reason, because in those days I was also motivated to do exercise, and so I was taking like a picture like every day, like not taking all the extra protein powders, like but just using regular diet, eating more protein rich diet, because if you're doing exercise you should. And then I was taking pictures, so I have those pictures, but I decided not to show it here.
Just a quick question, is it okay? I really would love to see your before and after calorie restriction. You should put it in your slide, actually.
Yeah, I think maybe I'll do that.
Actually, about the calorie restrictions, I have heard that there's a little consequence that yes, you look healthier, you look younger, but the one thing is about infertility. I have heard about, yes, the mouse with less calorie per day, that they're likely to have infertile compared to the normal calorie intake.
So I disagree.
What do you think about that?
Yeah, so I disagree with that, because I think obesity can make you infertile. So if obese people have less fertility. No. So see, there are two aspects, right? We are not talking about if you are a healthy individual and put you on calorie restriction. That is going to make severe complication. That's not what I'm talking about. So if you are obese, which like 500 million people in India are, maybe me too. If you put them on calorie restriction, you will see their reproductive health, mental health, everything is going to be reset. That's all I'm saying.
Manvendra, I'll just add it. What he said is very fine, but what you had apprehension is a true biological evolutionary reason. Let's forget for the time being his lecture. You know, the food scarcity is a trade-off during early time, when there is no food, nothing to eat, do you go for reproduction? No. So it's a biological trade-off during food scarcity, which we today call it calorie restriction model, blah, blah, blah. When there is no food, body has a strategy. It's a trade-off between reproduction and maintaining the somatic cells. So there is a very nice theory of aging, which is called disposable soma theory of aging, proposed by none other than famous Holiday, that Holiday model crossing over. He had proposed later on that we are endowed with fixed amount of energy. It's a bit philosophical and bit scientific. We are endowed with fixed amount of energy for two purposes. One is maintaining somatic cells. Another one is the reproductive cells. If you spend more energy on reproductive cells, reproduction, you are left with less energy on maintaining somatic cells. So the food scarcity during some times in famine or when there is a सूखा पड़ गया, कुछ नहीं हो रहा है, कोई खाना नहीं है, so that time your reproduction nature has stopped virtually, try to lower down, and there are evidences that severe calorie restriction does have that. But with the calorie restriction which he and I talking about is a mild calorie restriction which gives you hormetic effect, which is around 25 to 30%, not 70, 80 and 90% restriction. So that the both are correct.
Yes. So just to add on that, like now there was another study where they did like 20% calorie restrictions and 40%. So it's not like if you just keep increasing, it will get better. No. So if you do 20, that's good enough. 20 to 30, as Sir said, like that's good enough to see the changes.
Sir, can I have a question? Thank you for sharing your instinct on CVD. I have a general question. So just imagine there is a subject of the age that you have mentioned, like 40 to 45 years, male. That person has a great BMI. He works out every day. He's having every nutrition factors that he can have. And when we look at the blood results, all the other CBC and everything is fine. But when we look at the lipid profiles, we can see that there is a rise in the LDL, reduction in the HDL cholesterol, and a total cholesterol and triglycerides. So my question is, that person might have this familial or hereditary kind of diseases. So what can such person do to improve their heart health? Since this increased cholesterol level can definitely affect the heart health, right? So what can such persons do?
So I mean, like now the biomarker, like now what we said, like I mean, it was discussed before. So if you have a family history of anything, even cardiac, there's not much you can do. I mean, there's genetics, you have it, right? What you can do is you can try to take precautions, not to do what could be harmful for you. If you don't know, you are going to do what everybody else is doing, right? You see, like now Raju Srivastava died in doing exercise, right? Another film star died. Why? They may have a history, they don't know, nobody knows. Actually, the problem in India is, right, the major problem. We go to see a doctor when we have a problem. We don't go to see a doctor to see, can you check whether I am going to develop any problem in next 10 years? Right? So like my workplace, like we have a like, no, the reason I said like, no, you should go for test. We have a annual test, blood test, free of cost, okay, done by the institution. So you can't have an excuse. You just go there and then you follow routine, then go for the another one, right? And then if you see changes, right? So of course, these kind of things, if there is a family history, you should. And I would say like, no, in future, when I mean, already the genomics are very cheaper, right? Doing exome sequencing. And there are already established candidate genes for cardiac, for like no atherosclerosis, like no for liver. At least we should test those. And if you find like no mutation, which is going to significantly impact your health, then you should start following, taking precautions. No, so at least you are not going to make your situation worse by just like your own actions.
All right. Thank you, sir. Thank you.
Very nice presentation, Dr. Manvin. I think you should give some tuitions to the students because lots of questions are there. And as an example, you are here and sir rightly says that put your photographs here so that they can compare. Very nice presentation. Thank you very much.
Thank you.
Thank you, sir, for such an insightful session. Your research shed light on the emerging heart failure causes and its therapeutic approaches, and we truly appreciate your contribution to this field. As we conclude the technical session 6, I would once again like to extend my deepest gratitude to our esteemed chairperson, Professor Dr. Rajshri Ghosh sir, and our co-chairperson, Dr. Raj Kumar sir, for their invaluable contribution and guidance throughout this session. A heartfelt thanks to our distinguished speaker, Professor Dr. Sarita G. Bhatt ma'am, Professor Dr. Dinesh Yadav sir, and Dr. Manvendra Singh sir for sharing their knowledgeable and groundbreaking research, and to all our participants for their engagement and thought-provoking questions. Without further ado, I would now like to call upon stage Dr. Prerna Aditi ma'am to kindly felicitate Professor Rajshri Ghosh sir. Thank you, sir. Now I request Dr. Anukriti Raj ma'am to kindly felicitate our Dr. Raj Kumar sir. Thank you, sir. Now I request the Professor Dr. Rajshri Kumar Kaur sir and Dr. Raj Kumar sir to kindly felicitate Dr. Manvendra Singh sir. Thank you, sir.
Thank you. We now take a short tea break.